Hello, my name is Brian Filler, and I'm introducing the Tyngsboro High School 2012 iGEM team. We are composed of 10 strong, led by our teacher, leader, and mentor, Rebecca Revgala, and we are all ligated together by the common desire to combat carbon monoxide. At the beginning of this year, we designed our very own original logo with the help of Ashley Mayer, and we also made our own t-shirts that would unify us as a team. This is our very first year having an iGEM team, and we also had limited experiences with molecular biology and no experience with microbial techniques. We spent the first few meetings of this year talking about these topics, as well as using Tinkercell, which we later used to represent our genetic circuit. We also brainstormed our ideas of what we can use this season, and we chose for our project idea to create a cell that would release a banana output in the presence of carbon monoxide. The 2010 METU iGEM team designed a biobrick that expressed GFP and RFP in the presence of low and high levels of carbon monoxide, respectively. Our goal was to alter these biobricks so that instead of having RFP and GFP output, we would have a malodorous output. The problem with using malodorous outputs is that most malodorous outputs are not included in the registry, and most of them are also toxic. So what we did is instead we chose MIT's banana scent gene as our output because of its experience and availability in the registry. We also chose to use an indole-free chassis so that the scent of indole will not mask the banana scent. Now, why are we bothering building this genetic circuit? It's Massachusetts state law to have carbon monoxide detectors in your house, and also carbon monoxide is a colorless and odorless gas with a 250 times greater binding affinity to hemoglobin than oxygen, and it comprises oxygen transport from the lungs. As a result, 2,100 people die in the United States per year, and our goal was to try to reduce this number by creating our very own genetic circuit. Our ultimate goal was to create a cell whose smell would ring a bell. I'd like to introduce you to Taylor Vigno, who will be talking more about our genetic circuit.
Thank you, Brian. Good afternoon. I'm Taylor Vigno, and I'll be presenting the Circuit 2 design, which is our theoretical circuit called EcoFactory. EcoFactory came from the idea of genetically modifying E. coli to function as a carbon monoxide detector. When there's high concentrations of carbon monoxide present, a banana scent will be outputted. This design was a test, an olfactory test for the detection of carbon monoxide. And we decided to use an indole-free E. coli chassis for our circuit. And we decided to use this because we didn't want the indole smell, which is naturally produced by the E. coli, to interfere with the banana smell released in our circuit. Therefore, we're using this indole-free E. coli chassis to use a double transformation of two plasmids in our design of our circuit. Plasmid 1 consists of a single biobrick part, which is a device which is designed by the 2010 Turkey METU team, and this functions as a COOA transcriptional activator from the bacteria R. rubrum. R. rubrum is a bacteria that naturally metabolizes carbon monoxide, so therefore this part is going to be always producing the COOA protein and will be able to bind carbon monoxide to this protein in our circuit. Plasmid 2, this device is made up of a promoter, which is regulated by the LAC-I protein, and in its absence of this protein, it will promote the transcription of this coding sequence, which codes for the COA protein, and then it has the double terminator to end the sequence. In our E. coli chassis, our LAC-I protein will be absent, so we'll always be producing the COA protein in our cell. Plasmid 2 will be our second transformation, and to insert this in, we needed to follow the 3A assembly protocol to ligate these two parts, which this is the promoter that we chose from the 2010 METU iGEM team, and they use this PCOM promoter from R. rubrum to detect high concentrations of carbon monoxide. Therefore, we chose this part because we want this circuit to only run when there's high concentrations to warn someone of carbon monoxide. And then we'd be ligating this to the banana odor enzyme generator, which was designed by the 2006 MIT iGEM team, and to produce our banana smell. And this promoter has chloroamphenol resistance, and The banana odor enzyme generator has ampicillin resistance, so to follow the 3A assembly protocol, we would propose to use a kanamycin backbone for our third plasmid to ligate these two parts together. The promoter is the regulator, which would be binding to the banana odor enzyme generator. So in our cell, the CoA and the carbon monoxide will start the promotion for our circuit, and it'll be ligated to this part, which has the RBS and the coding sequence for the ATF1 gene, which is the enzyme that catalyzes the conversion of isoamyl alcohol to isoamyl acetate, and that is the banana smell. So in our circuit, when the ATF1 gene is made, it will convert isoamyl alcohol, which is present, to isoamyl acetate for our warning cell, for our carbon monoxide detector. Unfortunately, we weren't able to test or come up with any results for this circuit design because one of our parts was not present, this one, and it wasn't in the registry available to us, and also with our lack of lab facilities, we weren't able to use carbon monoxide, so we weren't able to test this circuit. But we came up with a proof of concept and an alternate circuit design which does not use carbon monoxide to test the functionality of the circuit design. And now I'd like to introduce Madison Vigneault, who will be presenting Circuit 1, which is our proof of concept design. Thank you.
Thank you, Taylor. Hi, I'm Maddie, and I'd like to explain the experimental circuit design. This circuit is composed of two separate plasmids, and the function of it is to safely test the design of the theoretical circuit without using carbon monoxide. In our circuit, we're using an indole-free chassis, and this is so the indole smell naturally produced by bacteria is not masking the banana odor generated by our circuit. The first plasmid being inserted into our circuit is the TET-R generator, which is made up of the constitutive promoter and the coding sequence for TET-R. The promoter is always on, so the TET-R protein is always being produced. Here you can see our first Tinker cell picture with the first plasmid always making the TET-R protein. The banana odor generator is the next plasmid being inserted into the circuit, and this is responsible for catalyzing the reaction of isoamyl alcohol to isoamyl acetate. This part is made up of the TET-R repressible promoter and the ATF1 generator. The TET-R protein is allosterically regulated by doxycycline, so when doxycycline is present, it binds to that, releasing it from the promoter and allowing for the transcription of the open reading frame downstream. In this case, it's the ATF1 generator, so the enzyme alcohol acetyltransferase 1 will be produced. This enzyme is responsible for catalyzing the reaction to make our banana scent. Here you can see our next Tinker cell picture with the second plasmid making the ATF1 gene. As you can see, doxycycline is the input, and it is acting like the carbon monoxide would in the theoretical circuit, as the TET-R protein is acting like the CoA from our theoretical circuit. Here's our final Tinker cell picture, where the isoamyl alcohol and the ATF1 enzyme is making the banana scent. Our protocol for this lab was adopted from the BioBuilder eChromI lab, and we chose to make a protocol have a double plasmid insertion, but not ligating any parts together. We weren't 100% sure the efficiency of our protocol, so we wanted to test it using PUC19, which is a commonly used cloning vector in E. coli that has an ampicillin selective marker. Our goal was to successfully transform PUC19 into indole-free cells on LB plates with ampicillin. Here, this plate shows our expected results with a plate covered in LB and ampicillin. This side would have a sample with transformed PUC19, and this side is transformed with no DNA. We would expect growth on this side because it has the plasmid with the ampicillin resistance, but because these cells have no ampicillin resistance, they would not grow. We performed the transformation twice, and we got the same results. And as you can see, DNA covers both sides of each plate on the PUC19 and no DNA side. We were not expecting this, so we decided to test further to see the error in our protocol. We decided to investigate our ampicillin concentration because that was the most logical issue with our protocol. We took many different plates with LB and ampicillin, and we used different concentrations of ampicillin from the same stock solution. And finally, we were able to get no growth by streaking indole-free cells with chloramphenicol resistance by using 10 milliliter ampicillin for 100 milliliters of agar, with ampicillin being from a stock solution that's 1,000 times concentrated. From there, we decided to start the transformation, the first transformation for building our circuit 1, and we just took the TET-R generator and transformed that into the indole-free chassis. As you can see, we were able to get some growth up here in the corner. Our next step would be to take those cells transformed and put them into the banana odor generator, but that was not successful. So to date, our results are inconclusive. And because we were only able to test the TET-R generator being transformed once, we're not sure of the reliability of those results. And still, we have not been able to successfully transform the banana odor generator, so we have not been able to construct our Circuit 1 design. In the future, if we are able to successfully build Circuit 1, our first step would be to perform a gel electrophoresis to see exactly what DNA is in the cell, and we would also perform other results, other tests to see our results. Next, I'd like to welcome Mike Sheets, who will be talking about those steps that we would take and overall our hopes for the iGEM Club in the future. Thank you.
Thank you, Maddie. Hi, my name is Michael Sheets, and I'll be talking about our future plans for our iGEM team and for our eco-factory project. To start, we definitely have a lot of room to grow, especially in research and development. This year was a big learning process as we came in with no synthetic biology experience, but now we know much more about synthetic biology and about the iGEM program. Most of our labs were also biobuilder supplied labs, such as Odor coli and E. chromi, to help us learn the techniques we need to use for our actual labs. For our first circuit, what we need first definitely is a successful transformation. To do this, we would need assistance from outside help who has more experience in microbial techniques, and we would also need more advanced equipment, and to do this, we would consider contacting a local lab facility or university to partner with them. For our theoretical circuit, the first thing we need, again, is a successful proof of concept, which would be our first circuit, so we know our eco-factory project has a chance for success. After this, we would need to transform METU Turkey's part that senses carbon monoxide and outputs COOA, and to do this, we would need to have it made, and we would need to find out at what cost and how long it would take to make this. For our second part, we would like to ligate together a promoter that is activated by COOA, and also with MIT's banana odor generating part. For this, we would consider transforming both parts in separately or ligating all the parts together to do one smooth transformation. For standardization, characterization, and computation, we would definitely want to work more on detecting the inputs and outputs for our system. For inputs for our first circuit, we use doxycycline, which we can adjust the volume to change the concentrations of doxycycline we use. For our second circuit, we would need to use carbon monoxide in an indole-free chassis, in a anaerobic chamber, so that we could. But for this, we would need to use something like an electric carbon monoxide detector that shows the percentages of carbon monoxide, so we can see at what levels our machine works best. For output, we would like to use our. We would like to test using a qualitative smell test and a fruit flies like Bible suggests for our first circuit. However, for our second circuit, again, it will be in an anaerobic chamber, so that will be more difficult to test. We would like to try to titrate the isoamyl acetate to better standardize and quantify our results. For next year, we have a lot of big plans for our team, and one of our largest is team member specialization, which would be that this year sort of everyone worked on everything, and next year we would want to have individuals who are more skilled in areas such as IT, public outreach, contacting businesses, and media. We would also consider having an iGEM support group of individuals who would be available when necessary to help us with areas that we struggled with, such as IT and programming. Next year, we would also like to do more efficient fundraising. This year, we already had multiple fundraisers and have recently sent out letters to businesses asking them to consider becoming corporate sponsors. However, next year, we would like to expand our fundraisers and do much more with that. For outreach and human practices, this year we already got an article in a local pamphlet and aired a commercial on our school news. However, we have even bigger plans for future years. Next year, we would like to do a DNA day at our high school where we talk to students about synthetic biology using fun games and hands-on non-lab experiments. We would also like to go to our local elementary school to do a day called Science Elementary, where we talk to the smaller students about science and especially biology and maybe even a basic level of synthetic biology. We also plan to present to our school committee in the fall about our project and our team, and would like to contact our local fire department about local carbon monoxide statistics and emergencies. We also plan to next year to put out an app and another video. Next, I will call up Kathleen Barrett, who will talk about our experience this year with iGEM. Thank you.
Hi, I'm Kathleen Barrett, and I'm going to talk about our experience this year at iGEM. This is our first year in iGEM, so we ran into a few problems. In the beginning, we struggled to come up with a feasible idea. We found that we'd come up with something that was really interesting, what we thought was really unique, and after putting a little bit of research into it, found that some other previous iGEM team had already completed the project. So after a lot of effort and work, we decided to combine one of our ideas with the work of a previous iGEM team to come up with a unique system to solve a different problem. Our next issue is that we had little to no previous experience with microbiology, and we thought this was going to be our biggest obstacle to overcome. However, the BioBuilder experiments and the iGEM headquarters and the iGEM website were wonderful resources in learning about microbiology, and we were able to actually rather easily overcome this obstacle. Our next problem was our access to equipment. Because we're from a rather small school, we don't have some of the more expensive equipment needed to complete many of the experiments done in synthetic biology and microbiology. Fortunately, we were able to find modified versions of protocols that allowed us to complete our experiments, and we were able to design our own versions of the equipment. For example, we made our own centrifuge out of a salad spinner. Our final issue was that we had limited funding. We are an unfunded club, so in order to raise funds for our club, we had to do a lot of fundraising on our own. We were very fortunate that we had a lot of individuals and companies that were willing to donate their time and money to our clubs, so we were able to proceed. Overall, we had an amazing experience with iGEM. We learned a lot about the techniques of microbiology and synthetic biology. We got hands-on experience in the lab that we would not have been able to get in a normal classroom experience. We were able to work together as a team and learn how to think for ourselves and to implement and execute our own unique ideas. This was an amazing experience, and we're very thankful. This concludes, we'd also like to thank numerous companies and individuals for their support and their donations of time, money, and effort. We'd like to extend a special thank you to Alyssa Hennings from Ginkgo Bioworks, Natalie Kudel from MIT, and Kitwa and Megan from the iGEM headquarters, whose time and effort they put into helping us was essential to our club. This concludes our presentation, and we'd like to thank you for taking the time to watch it.
Go iGEM!
Okay. It's awkward because the cameras aren't off yet.